Hello, biology, and welcome back. Today we're going to start talking about Chapter 10, which the stuff in Chapter 10 is all about DNA, RNA, and protein synthesis. We're going to talk about how the code of DNA actually becomes useful. How do we get that code of DNA, that genotype that we have, and how do we turn it into a phenotype? So how do we take our genetics and turn it into what an organism can actually use? Let's jump right in. The first thing we need to talk about is the discovery of DNA, because we don't have a long-standing history of DNA. DNA hasn't even been discovered yet at the time of this video for 100 years. So let's talk about how we came to discover DNA and how we came to know its role in the body. So first we're going to talk about is Griffis experiments. And in 1928, this scientist named Frederick Griffith was studying Streptococcus pneumoniae, which is this guy right here. Strepto means it's in long chains, and coccus means that it's these round kind of structures. In mammals, Streptococcus pneumoniae is a disease that can cause lung disease, pneumonia. And when a disease is able to cause disease, we call it virulent. It has the ability to make an individual ill. Something we need to talk about to understand Griffith's experiments is that bacteria have the ability sometimes to have capsules. And capsules are these kind of surrounding the bacteria made of sugars that the bacteria makes. It provides protection from the environment. So the one over here, this one on the right, is a capsulated form of this same bacterium on the left. This one on the right here, right above my head, is the virulent strain of bacteria. It had smooth edges on the outside, so we called it the S strain. The non-virulent lacked the capsule and grew in rough colonies, or we call the R strain. S strain, virulent, causes pneumonia. R strain does not. So what Griffith's experiments actually looked like is he did a series of 4 different tests to test the 2 strains and how virulent they were. And in the first one, he injected mice with the rough cells, the non-virulent ones. The mouse lived. Check. In the second one, he injected the mice with the live S cells, those smooth ones, those virulent ones. And in that one, the mouse dies. He then took these S cells and he killed them. He used heat, and he basically, heated them up and killed the bacteria and injected them into the mouse. And in this one, the mouse lived. And then he killed the S cells with heat. He mixed them with live R cells and then injected them into the mixture, and the mouse died. So what does this all mean? Well, the conclusions of Griffith's experiments was that the heat-killed bacteria released a hereditary factor, and we talked about that when we talked about Mendel. Something that these bacteria have allowed them to kill mice. But when they were killed with heat, these materials got expelled into the kind of whatever medium that these other bacteria were in, and it transferred this ability to cause disease to the non-virulent strain, or the R strain. When we have genetic transfer of genetic material from one cell to another cell, it's called transformation. And that's what happened here, is those rough cells were transformed into smooth cells. And this was all very exciting, and Griffith was very, accomplished on this. But he didn't know what this genetic factor, this hereditary factor, was. And in the early 1940s, another guy called Avery Oswald decided to carry on Griffith's experiments. And so he tested where Griffith's factor was DNA, RNA, or protein. Because we knew that these things existed, we knew that there was different molecules we could pull out of cells, but we didn't yet know which one was carrying the actual information. So he would use enzymes that targeted each of these substances and broke them down, turn them into non-functioning versions of themselves. He would then mix the bacteria with live rough cells and inject that back into mice. So, the conclusions from Avery's experiments: when he damaged the proteins, they transformed and killed the mice. When the bacteria with the damaged RNA was used, they also transformed and killed the mice. And then on the final one, when he did it with damaged DNA, the mice did not die. The bacteria did not transform, so the mouse got to live. So what Avery's experiments showed is that DNA was responsible for the transformation of these bacteria to become virulent, to have that ability to kill the mice. Moving on to 1950s, we have the Hershey Chase experiment. And in 1952, there was a gal by the name of Marcia Chase and a guy by the name of Alfred Hershey. And they used these little guys here, which are bacteriophages. They are viruses that infect bacteria. And yes, they do look like that, little spaceship looking guys. What they were able to do is they were able to take radioactive isotopes and label DNA and the protein that went into these little phages, into these little guys. And what they discovered is that when these guys attack other cells, they inject very little protein, but all of the DNA that's inside of these little capsules here gets injected into the cell. So we knew that bacteria use DNA, but now we found out that even viruses are using DNA to transfer hereditary material. So let's talk about the structure of DNA. And the two famous people we're going to talk about there, plus one, are James Watson and Francis Crick. And they set out to discover this, the shape of DNA. And they discovered that it makes this double helix, or looks like this winding staircase where you have one staircase over here and one staircase going the other way. And then all of these are like rungs on this ladder. But they, even though they're credited with solely finding this work originally, they relied on other scientists' work like Rosalind Franklin, one of our strong women in STEM in a time when it wasn't cool to be a woman in STEM. And she took X-ray photos of DNA crystals, and this is what they look like right here. And you can kind of see this here. This is a picture of this in, a two-dimensional format. Francis and Crick won the Nobel Peace Prize for discovering this structure, but we have since started to recognize and worldwide the acclaim of Rosalind Franklin and her contributions to this kind of discovery. Because without her work and her X-ray pictures, Watson and Crick probably wouldn't have been able to get to the double helix structure. All right, let's talk about the individual nucleotide of DNA, because DNA is a nucleic acid. It's made of two long strands, but each strand is made up of just many, many parts of these. And these repeating nucleotides each have three parts. They have a 5-carbon sugar, which in DNA is deoxyribose. They have a phosphate group, high-energy phosphate group here at the end. And they have a nitrogenous base over here. In this case, this one's adenine. But the nitrogenous base contains nitrogen, carbon, and it accepts hydrogen ions to make some nice hydrogen bonding. So here's the individual nucleotide. Here, it's got several bonds within itself, but there are bonds that hold the entire DNA molecule together. Sugar and phosphate kind of form this backbone of DNA where they stair-step. Sugar, phosphate, sugar, phosphate, sugar, phosphate. The nucleotides form the rungs of the ladder. They're covalently bound to their sugar, and it takes 10 DNA molecules to make one full spiral around. Now, there are 4 types of nitrogenous bases, and they're facing towards the center of the DNA molecule. This one is facing this way. This one is facing this way. And they form hydrogen bonds, which are these blue lines here in the middle, with other bases on the other strand. Some of them form 2 hydrogen bonds, and some of them form 3 hydrogen bonds. So there are 4 kinds of base, and we're going to talk about base structure here on this slide. And the 4 kinds of bases are thymine, cytosine, adenine, and guanine. And we'll abbreviate them as T, C, A, and G. Adenine and guanine have 2 rings in their structure. They're what we call purines. Thymine and cytosine and uracil, which we'll talk about in an mRNA, have a single ring and are called pyrimidines. In 1949, we have Erwin Chargaff, and he creates this kind of theory and hypothesis about complementary bases. And what he found out is that the percentage of adenine equals the percentage of thymine, and the percentage of guanine equals the percentage of cytosine. Or basically, the amount of purines equals the amount of pyrimidines. And what this creates is it creates these rules, these base pairing rules, where cytosine on one strand bonds to guanine on the other strand, and adenine on one strand always bonds to thymine on the other strand. And what this is this complementary bases is the base pairs that always bond to each other. So A will always bond to T and vice versa. And C will always bond to G and vice versa. So base sequence is when we have 2 strands of DNA, there's always the same number of bases on each strand of DNA. So if we're on one strand of DNA, the bases are A, C, C, G. The complementary bases are T, G, G, C. When DNA is replicated, one strand will be the blueprint for the opposite strand. So they will both be able to make their own strand. If we took A, C, C, G here, we could write the complementary strand T, G, G, C. And the same thing down here. If we take T, G, G, C, we can make a complementary strand of A, C, C, G. All right, so DNA molecules, for ease of access and because our brains work better, we will draw them flat like this sometimes, and we'll even break it down simpler and do T, G, C, A, G, T, etc., etc., etc. Where we just take this nucleotide and then draw it just in a straight line. It helps our brains, instead of trying to think about it in a spiral form, to just simplify it and put it in a straight line. All right, so now we're going to change gears and start talking about DNA replication. So in DNA replication, this is the process by which DNA is copied in a cell before it divides. So we do this, we get DNA replication before mitosis, before meiosis, and during binary fission. And during this, the 2 strands will separate, like they are here, and each one of them will be copied. And the yellow in this is the copied material. As you can see at the end here, each DNA molecule is half blue and half yellow. It has a new strand and an old strand. All right, step 1 of DNA replication. This enzyme here is called DNA helicase. And it moves along the DNA and it breaks these strands apart. It splits these H bonds in the middle here. This Y-shaped region here, kind of upside down shaped Y, is called a replication fork. This is where that DNA is being split. In step 2, we're going to use DNA polymerase, which is going to, it's right here, and right here. DNA polymerase is going to add complementary nucleotides to each original strand one at a time. So if the strand was A, C, C, T, that polymerase would come through and it would go T, G, G, A, like that. It will make covalent bonds between the new nucleotides and the deoxyribose sugar on one side and the phosphate on the other to create that nice sugar phosphate backbone that will hold all of this together. In step 3, the polymerase finishes replicating and the DNA falls off. Excuse me, not the DNA, but the polymerase falls off. The result we get is 2 identical molecules. And each new DNA molecule, one strand is new DNA and one strand is old DNA in a process called semiconservative replication. So that's the one in the middle here, was the semiconservative, where each one of these gets a blue and each one makes a new red. And then when we make a new strand, you get half and half here. For conservative replication, which was another proposed model, the DNA would be used as a template and then put back together. This isn't how it happens. And in this one, dispersive, the idea was that chunks come and go and come and, they use one chunk here and one chunk there and one chunk there, and then it ends up getting all messy. And that doesn't happen either. The one that is super important, the one that our bodies use and all DNA uses, is this semi-conservative here. All right, so at the replication fork, DNA synthesis occurs in different directions. One strand is going in the same direction as the replication fork. As this fork moves this way, this guy is moving in the same direction as it. And so it synthesizes DNA in a long line. The other one is moving against the direction of replication. And what happens is each of these little chunks of DNA get synthesized on their own, and these are called Okazaki fragments. These will later be glued together using DNA ligase. Now there's some difference between replication and DNA synthesis in prokaryotes and DNA synthesis in eukaryotes. In prokaryotes, they have one circular chromosome. Replication is beginning at one place, and it will continue until it goes all the way around the circular chromosome. Just one replication fork. For eukaryotes, they have these long, rod-like chromosomes. If we were to replicate them the same as prokaryotes, it would take us around 53 days per chromosome. So what actually happens is replication happens at many different points. There'll be many different replication forks along the line here. At each point, it's going to go both up and down until it runs into the next replication fork, and then so on and so forth until the whole chromosome is replicated. All right, DNA errors in replication. So there's only about one error every billion base pairs. So the same if you're going to type up your biology textbook or a similar textbook 1,000 times, and if you only made one spelling error. The nice thing is even though these odds are really low already, DNA polymerase has repair enzymes that proofread the DNA. And they'll take out a mismatched base pair. Let's say you put in a T instead of an A, and it will add the correct one. It'll put in that A, and it'll change that so that you have the right base pair in there. When we do get a mismatched pair or we get something wrong, it's called a mutation. And this is the change in the nucleotide sequence of DNA. So in this example here, this T has been mutated into a C. So thymine has been mutated into a cytosine. And what this does is it disrupts gene function. And some things that can do this are things like sun, things like radiation. Anything that is a mutagen, meaning it causes a mutation, has the opportunity to cause mutations in healthy normal DNA. DNA replication has the opportunity to happen in cancerous situations. We can get mutations in the genes that control cell growth. Remember, we had 3 different checkpoints that prevented cells from rapidly and uncontrollably dividing. But if there's a mutation in those genes, then the cell starts to divide uncontrollably and causes something like this, a mass, which we call a tumor. Tumors can either be benign or malignant. or malignant, depending on whether they are spreading or dangerous. But a lot of research going into DNA replication is particularly of interest to cancer researchers because they want to learn how to prevent this from happening. All right, the last part of this chapter is protein synthesis. So we have a DNA sequence. We need to turn it into a protein that creates a phenotype. So we have a genotype. How does that get to the phenotype? So DNA is the blueprint for this process. When we're looking at something, we're going to look at a specific gene. So this specific gene is on a chromosome here. This is one protein that it's carrying information for, this pigment that makes purple. So protein synthesis is how we express our genes, how we turn genotype to phenotype. Another type of protein are enzymes, like these ones here, hemoglobin, which you use in your bloodstream, antibodies, which you use to fight off infections, and collagen, which is used in all your hair and muscle and everything else. They're all types of proteins. Other proteins are biological catalysts that regulate cell, chemicals reactions in the cell, and all of your bodily functions require catalysts. So the catalysts that you have, the enzymes that you have, is coded for by your DNA. So we talked about the building blocks of DNA. We talked about nucleic acids, but one chunk of DNA is going to have lots of different bases on them, or triplet codes, to code for one amino acid using mRNA and tRNA, and we're going to talk about that process. And those amino acids will then build together to form a singular protein. So examples are AAA, adenine, adenine, adenine is a triplet that codes for UUU in mRNA, which makes the amino acid phenylalanine, and CCT codes for GGA in mRNA, which is glycine. A single gene will have between 300 and 3,000 base pairs, so between 100 and 1,000 amino acids. So the role of RNA is as a messenger and decoder for DNA. Remember that inside the cell we have the nucleus, and the nucleus has those pores. We need to get the DNA signal out of there into the rest of the cell. So some things that are different about RNA is it uses ribose instead of deoxyribose, and it is single-stranded instead of double-stranded. So the single-stranded RNA is able to move outside of this pore into the cell. Another important thing is instead of the base thymine, RNA uses uracil. So you see uracil here instead of thymine. DNA uses thymine, RNA equals uracil. So in RNA, A will bond to U, and U will bond to A. There are no T's in RNA. There are three types of RNA. We've learned about these before, but it's really important in this chapter to understand that there are three types and how they're used, so we're going to talk about it again. There's messenger RNA, which carries the message in this single nucleotide strand here. So it carries a signal from the DNA to the ribosome. There's ribosomal RNA here, which forms up the ribosome, which job is protein synthesis. And then there's transfer RNA over here, these clover-shaped, here's the clover shape here, molecule that brings amino acids to the ribosome to build them one at a time, these little amino acids on the top here, to build proteins. There are two phases to protein synthesis. There's transcription, where we take DNA and we make complementary mRNA here. This brings the signal from inside the nucleus to the outside. We talked about DNA can't move through those nuclear pores, but RNA can. And then in the second part, translation, the information from this mRNA here is decoded to assemble proteins. So we're taking this RNA and we're making it into protein. So let's talk about transcription first. In transcription, we take RNA polymerase, which forms RNA chains based on a DNA template, and it starts at what we call a promoter. There'll be an area here where it says, hey, right ahead of here is the gene that you're looking for. So it allows this attachment of RNA polymerase, and it starts there. The same way that DNA was added one at a time, complementary RNA bases are added one at a time. Usually, the start codon is AUG, methionine, and we're going to talk about what a codon is next. So the transfer of information from DNA base sequence into a complementary base pair of mRNA. In DNA, these triplets are called triplets. When we turn them into mRNA, they are called codons. So this A turns into a U, T turns into an A, G turns into a C. It's complementary, the same way as when DNA is being synthesized. On your assignment, you'll see things like this. You'll need to know how to use them, so we're going to real quick give you a how-to on it. If your DNA code is ACG, that means that your RNA code will be UGC. If it's CTA, that means it's going to be GAU. If it's ACA, it's going to be U, because remember there's no thymines, GU. And if it's CCC, it's going to be GGG. Now, to use this, we're going to take these codons. I'm just going to real quick draw some lines here so we got a little bit of. These are the codons here. We'll take the first one, which is U. You'll go to U on the first one here. And then the second one is G, and you'll go all the way over to G, UG. And then the third one is C, and you'll go UGC, which is cytosine here. Or cyto. We're just going to call it cyto, because we don't have enough room. But cytosine would go there. That would be the amino acid there. So you'll take the first base here, you'll get the second base from this one, and you'll get the third base from this side. All right, so the second one here, C, and then A, CA, and then U, CAU, histidine. Hist. Our next one, UGU, cytosine again. And then our last one is GGG, GGG, which is glycine. So we took this DNA, we turned it into complementary mRNA, and then we ended up with the amino acid sequence. And this is kind of how you use these. Now, it would be very impractical for our DNA to use every single chunk of DNA when we're trying to get one gene. When you're in the liver and you're trying to make bile, which helps you break down fats, you want to just make bile. You don't want to also make eye cells and eye coloration proteins and all that things. So we use these termination signals to stop the translation and transcription of DNA. So it marks the end of a gene. When RNA polymerase reaches this, it unbinds from the DNA and falls off. So there are three stop codons, or three nucleotide sequences that stop RNA transcription. And they are UAA, UAG, and UGA. And they are kind of all located right here, are the stop codons. We're going to talk about this one here, AUG, the start codon, next. Excuse me, we already talked about the start codon. So we're going to start talking about translation now. Translation in mRNA will take its codons, and this is the green line here, to the ribosome. tRNA then brings the amino acids to the ribosome to match up these codes that's on the mRNA codons. For amino acids, there's 20 common types, one for each amino acid. There's 20 tRNA types. They have different 3-base sequences that attach to specific codons. So their 3 kind of code sequence is called an anticodon because it's the opposite of a codon. And then the last thing we're going to talk about is the human genome. It's the genome of any organism. It's its entire genetic content. For humans, there are 30,000 genes that we know of. There's probably more. And there's 3.2 billion base pairs of DNA nucleotides. So what that means is there's actually 6.4 billion nucleotides in a human body, which is incredible. You have a lot of genetic information. We're still looking at and discovering a lot of it to figure out what's even in there and what's it all used for. I hope that you enjoyed this video. Thank you for watching, and we'll see you in the next one. Ciao, ciao.